you will learn about Chapter 8, Recombinant DNA Technology. In this chapter, there are 3 subtopics, which are 8.1, Recombinant DNA Technology, 8.2, Method in Gene Cloning, 8.3, Application of Recombinant DNA Technology. At the end of this video, you should be able to define recombinant DNA technology. You should also be able to define and explain the 5 tools used in recombinant DNA technology, which are: first, target DNA, also known as gene of interest; second, restriction enzyme; third, DNA cloning vector; fourth, host cell; and the last one is modifying enzyme such as DNA ligase.
What is actually gene technology? It is the various techniques for sequencing and manipulating gene DNA. Manipulating genes means modifying and recombing DNA to produce desired products such as proteins or animals and plants with desirable traits. One of the main gene technology is recombinant DNA technology. Recombinant DNA technology is a technique that joins 2 DNA molecules from different sources to produce new genetic recombination. It is commonly used in various fields in science such as medicine, agriculture, and industry.
There are 5 tools needed for recombinant DNA technology. The first one is target DNA, also known as a gene of interest. In this gene, selected to be cloned. And the second is restriction enzyme, which is used to cut DNA into fragments. The next tool is DNA cloning vector. Its function to carry the target gene into the host cell. The following tool is host cell. It allows the cloning vector to replicate such bacteria cells. The last tool is modifying enzyme, which is function to join DNA fragment together such as a DNA ligase.
Okay, in this video, you should be able to describe the gene of interest and the characteristic of host cell. As seen in this figure, genes of interest is a fragment of DNA which is part of a larger DNA strand that is in target cell. For example, the genes of interest for insulin is located in the pancreas cell. In order to replicate the target gene, the host cell is needed. It works by carries cloning vectors that contain the target gene, also known as recombinant DNA. Host cell is an organism that has been used to receive recombinant DNA for cloning purposes, such as E. coli and yeast. There are 4 characteristics for an organism to be a host cell. First, it must be able to accept the recombinant DNA through the transformation process. Second, it must be able to maintain the structure of the recombinant DNA from one generation to another. Third, it must be able to amplify the gene product from recombinant DNA. Lastly, it must be able to express the clone gene.
In this video, you should be able to explain restriction enzyme, an example of enzyme that produce sticky ends. You should also be able to explain modifying enzyme and its function, such as DNA ligase and Taq polymerase. Restriction enzyme is naturally found in certain bacteria that are resistant to bacteriophage infection. It is function as an enzyme which that recognize a specific DNA sequence called recognition site, and then it will cut at a specific point on the recognition site.
There are various types of restriction enzyme. Different restriction enzyme recognize and cut at different specific restriction enzyme. This produce DNA fragment with sticky end or blunt end. The receiving site is a sequence that is consists of the same 4 to 8 nucleotides on both DNA strand but arranged in opposite direction. 5 prime A G C T T 3 prime reads, while the opposite side is 3 prime T T C G A 5 prime. The sequence of receiving site has palindromic sequence. Palindrome means the base sequence of one strand reads the same as in the opposite direction of complementary strand of DNA. For example, as shown in figure here, the function of palindromic sequence is to act as a recognition site for receiving enzyme and then cut at the receiving point on both DNA strand.
Restriction enzyme is divided into two main types based on the technique of cutting, which are either staggered cut or straight cut. When staggered cut is used to cut DNA fragments, it will form sticky ends of DNA fragments. While if the DNA fragment is cut straight through, it produces the blunt end of the DNA fragments. The table shows the example of restriction enzymes and the organism that produce the respective enzyme. So let's look at the first enzyme, EcoR1, produced by Escherichia coli. So at the restriction point G and A, there will be a cut at both strands. Thus, this will produce sticky ends. Another example of sticky end is BAMH1 enzyme. Okay, so here the restriction point will be between G and G on both strands. While in enzyme SMA1, it will produce blunt end. Why? Because it will cut between the C and G at the restriction sequence.
So now we need to identify the bacteria that contain the recombinant plasmid. So a screening process is required. So E. coli colonies are cultured on a medium with antibiotic ampicillin. It is used to detect the bacteria containing recombinant plasmid and eliminates the bacteria without the plasmid. This is because the transformed bacteria with recombinant plasmid contain ampicillin-resistant gene. So, only bacteria containing plasmid that have antibiotic-resistant gene can survive and grow in the medium. So, as shown in the diagram here, okay, can you see it? So, as I said before, bacteria with plasmid will only survive after 15 or 16 hours, while bacteria without plasmid will die or be eliminated. So after that, the survival bacteria colonies undergo further screening, which is called blue-white screening. The survival bacteria colonies is cultured on medium with agar, which is white in color. The agar is the substance similar to lactose that helps to identify the plasmid that carries the target gene. The bacteria colonies without the target gene will turn blue in color. So remember, without target gene, turn blue in color. So this is because the LAC-Z gene is still functioning. So if the LAC-Z gene is still functioning, means it can produce beta-galactosidase, which hydrolyzes the agar from white color to blue color. So that is why the colonies or the bacteria without the target gene will turn blue in color, while the bacterial colony with the target gene remain white in color. Here, the LAC-Z gene becoming non-functional or not functioning. This is due to the presence of target gene inserted in the LAC-Z gene region. So when the target gene is inserted in that region, so the bacterial colony lack the beta-galactosidase, meaning it will not produce the beta-galactosidase. So the white colonies show bacterial clones which carry recombinant DNA clones. So as shown in the diagram here, so as you can see how blue-white screening works. The plasmid without insertion with gene of interest will turn blue in color. So without blue, okay, remember. So blue due to the LAC-Z is still functioning, okay. So without blue functioning, so that is the keyword. So while the plasmid with insertion, okay, of the gene of interest in the LAC-Z region maintains white in color. So why? Because the LAC-Z is disrupted or not functioning.
Okay, first step is isolation of gene of interest from donor DNA and plasmid from bacteria. Second, donor DNA is cut twice while the plasmid is cut once by the same precision enzyme. Third, gene of interest is inserted into the plasmid and then ligated by DNA ligase producing recombinant plasmid. Fourth, the transformation process occurs when the recombinant plasmid is introduced back into bacteria. Then, the bacteria is replicated through amplification process, forming bacterial colonies. At the end, the bacterial colonies must undergo screening process to identify the desired bacterial colonies with the gene of interest by ampicillin screening and blue-white screening.